All right, so let us start this roundtable. The title of the roundtable is here, and I'm going to comment on this title in a minute, but before that, I would like to introduce the panelists. You all know Paola Castagnoli. She has given a superb talk on dendritic cells. She's the scientific director of SIGN in Singapore, and she has been very, very instrumental in building the whole place together with many other colleagues who are here today. Catherine Fridman is an immunologist, and she has a special function in University Paris 5 in teaching or in governing the teaching to students. So this is particularly appropriate for that purpose. Jean-Michel Claverie is an old complice, as we say in French. In 1986, we published the so-called peptidic self model. Since he diverged entirely out of immunology, I think I can say this. I must say it's probably all the same, that he's one of the most provocating person I've ever met. So I'm sure he will be, and he's very pleased for that compliment, that you can guess. Okay, so let us start. Let me just explain to you what I would like to see happening in this roundtable. And if there are a few students who are interested to participate, if they could come, and many show themselves, we would like to discuss with them as well to know their expectations. Because what we should teach to the young people is really the issue of how immunology will be in 10, 20 years from now, and therefore how should we train the students, PhD students, of course, not young kids, as Catherine thought for a minute. How should we train them in order for them to be, at in the best possible situation to run the research and the science. In 1999, I don't know if you read that, there was a very, very good paper in Nature by Hartley and colleagues, which basically advocated for modular biology. This was one of the first, in my view, one of the first well-conceived papers on what's going to happen in systems biology, modular biology after molecular and cellular biology, meaning that the relevant subset in terms of complexity was basically the issue of the modules. The conclusion of that paper is that it was really time to try and teach the students completely differently to be able to be confronted with the complexity which was raising up. I must say 13 years after that paper, I'm not sure it had an enormous impact on that particular basis, even though, as we will certainly hear, there has been shifts in the teaching and in the training that go into that direction. I will comment just to frame the debate, and then the panelists will talk. I am struck by a variety of issues which I would like to share with you very briefly. One of them is that there is a loss of the historicity in the whole system. It's not only true in immunology, but basically people do not know the history of science anymore. And basically, if you look at any paper, or most papers, it's rare to find references which are more than 3 or 5 years old. Actually, the history is not usually there. Now, this is a problem, of course, of edition, time, so on and so forth. You're not allowed to have more than that many references. Still, the system is such. My second point is technicity. It's really striking to see what's going on. And a week ago, I heard a talk by the chairman of the Beijing Sequencing Institute, and he was quietly explaining that they are sequencing 100 human genomes per day. and that his small and modest operation employs 5,000 people. So, well, this is coming. It's just an illustration of the enormous amount of data which are now acquired because of the new technologies. It's not only sequencing, of course, but sequencing is so far the most productive in terms of giving you enormous amount of data. And so the question is, what are we going to do with this explosion of data? And we have more data that we can handle, and I think this is really an issue. Actually, this issue was underlined by Sidney Brenner and many others, but Sidney Brenner 7 years ago already underscored the problem that the issue was not to get more data right now, but rather to understand what we mean, which I absolutely entirely agree with. So that complexity is linked to what I call systematic immunology, that is the acquisition of lots and lots of data in a systematic fashion, which has some virtue. And it's a sort of a prerequisite for systems immunology, which is something relatively different in the sense that here you need models, you need some intelligence in order to build models and to test the models. So it's a different kind of activity. So I think there are real issues and conceptual issues in coping with complexity as it is showing up. Another point which is dear to my heart, not my heart, my reason, is demography. I was sharing with some of you that I calculated the proportion of the French population involved in research in biological sciences, in life sciences. The proportion is 0.1% of total French population, which means 0.2% of the active population. That's a lot of people, okay? Now, this proportion is probably about the same in most developed countries, a little more in the United States maybe. But the point is that the total number of people in research in life sciences is going to double or triple in the next few years because of China, India, Brazil, and the others. Now, in my own view, and I cannot really prove that figure, the degree of redundancy of what's published in the literature is at least 5 or 10. That is, the same things are published at least 5 or 10 times. It's understood that you have to reproduce the data in order to be sure that they are true, so that's understood. So that probably allows for redundancy, a factor of 2, 3, 4, 5. But 10 is a lot, and if the number of people double, and if the redundancy index gets 20, I think we have sort of a problem. Notwithstanding the fact that the competition is raising, of course, for that very same reason, and that we have a problem on that side. Now, that will impact, is already impacting, and will impact all sorts of things. For example, communication is more and more difficult in my view. It's becoming more and more local. So you have big meetings in the US, you have not so many Europeans anymore. Same in Europe versus the US and Asia versus and so on and so forth. Communication is not exactly the same as it was when Jim Watson was running Cold Spring Harbor. It's changed entirely. And so now the publication system is getting extremely complicated and difficult, and I must say, in my own opinion, very often unfair. So I think this increase in volume has to be thought about and can become problematic. And I will finish with a word which may look a little philosophical, but after all, science is a democracy. Actually, it's the best example of a democracy because we discuss together, we tend to agree, sometimes we fight, but by and large we agree. We have a fantastic freedom and we are really able to handle a lot of our own affairs. And I think we do not really think so much about these changes, and I would advocate for thinking along these lines or the other lines that the members of the roundtable are going to put on the table, maybe in more pragmatic terms. But I think there are all sorts of governance problems in the system. For example, in my view, if you take the microarray data right now, I guess 90% of what's published is not usable because things are not done along the same standards. So what we call now modern biology or systematic immunology or whatever requires a social organization, which we see coming, of course, because the sequencing people are better and better organized and so on and so forth. Nevertheless, this is going to increase more and more as more data come along. And we have to get used to the fact that the sharing of the data in an appropriate format is going to be a major condition for science, and that it does require a certain mindset, because if you believe too individually, you will not comply with this, and the system will not be optimized in any way. Apart from that, of course, you have ethical problems related with the state of competition, even in the publication system, as I mentioned, and by and large with the interface with the private world, which is absolutely necessary, but has to be framed in a proper way. So what I mean is that I think that in front of us, there are a lot of questions which need to be addressed, or should be addressed, or at least thought of. And this is why I thought that this roundtable would be interesting to conclude this meeting. So then I will leave first Catherine, then Paola, then Jean-Michel, expressing themselves. And again, probably in more pragmatic terms than I've been doing.
So I put here some just those two ideas and principles which seems to me very important for young students who enter into research. For those who choose the route to research, I think, and these principles are, I think the same today as they were many years ago. It's to have an open and innovative mind. Science is moving very fast, and now we have to face two systems, immunology. But I remember Philippe 20 years ago when we had to face to molecular biology. We went to your lab to learn molecular biology, maybe 25 years ago or something like that. So, well, we have to face always in our life of scientists to new challenges and new technologies. Now systems biology is a new challenge, but there were others. And so we need to learn all our life long new things, new techniques, new ideas. I think also it's important to teach the students of refrain from doing me too studies. You see what I mean? Is to copy something which has been published somewhere in another species, another, let's say, I think it's very important to be innovative and to go far from, let's say, classical roads. And if you think you are right, pursue in your direction. And of course, it's important to reproduce new data, to have them reproduced by other groups, but let's be innovative. And I think it's quite difficult in the present system, where one asks to the, let's say, for instance, French agencies and everywhere, students are faced to the problem of publication. They need to have publication lists in excellent journals. And so it's something very difficult to be, because innovation does not always mean to have quick data. I know, for instance, Jérôme Gallon in our lab, who has been doing very innovative things in the field of T cells into tumors. He has been during 4 years without, let's say, a tremendous publication. This was good because he was in the French system with a tenure position, but how would it be if it would be in the present system where there is this very strong pressure to publication? So it's not always easy to be innovative. And also should be open to new ideas and not to be frightened and pursue research. I think also research is, we live in a society, so these students should learn to work in groups. They should learn to communicate, as Philippe said. Our job is something to do with communication, and teaching also has to do with communication, and I think it's very important, this. Networking also is important for students, that they become member of network, and they keep contacts with group of people and keep contacts with their mentors. That's also, I would say, something important. And this, but not last, choose a good lab to do their research. So then I address the question, what do universities teach during master studies, and do the doctoral school do into these several items? I think universities, they can learn about basic principles about the immune system. Now they learn to work in groups. This is especially in second year of master, but also first year of master. They do together literature analysis. They write scientific project based on analysis of the literature. They learn to communicate because they present their work among other students and professors. They learn to discuss, but I would say, in my experience, in France, there is a loss here, because when you go to European schools, you see many students from England, from Italy, from Germany, interacting very easily with speakers, addressing questions. But the French, I noticed that the students from our French universities have still things to learn in that area. So I think we should push them, and we are trying to do that at Paris 5, at Paris 6, but I think this is very important. Then what do the doctorate schools do? As you know, here it's the teaching system for PhD is separated into doctorate schools. I think with the new system, they don't have, let's say, much to learn, let's say, in academic terms during the doctoral school. And it's really, during 3 years, I think it opens their mind. They start networking because they have this network with other students of the doctorate school. And I think, at least in Paris, but I guess in other big cities also, the doctorate schools help them to plan their future. Because it's clear that among the PhD students, only a small part of them will enter into research. And so then it's to show them that research is not the only option. There are many others. And they can be trained in these other options during their PhD. Then what do the labs do? So this is much more difficult to answer to this question, because it's very heterogeneous, of course, from lab to lab. Because I think that our work is still something like artisanat. I don't know the name. There is some kind of compagnonnage. Hein? Voilà. And, pardon? Cottage, yeah. And it's some kind of compagnonnage during which occurs between mentor students, and this is very heterogeneous and depending, of course, on the lab and the mentors and the team where they work. So this is what I could put, but I don't know. If you have questions or reactions? But regarding Philippe's point, I think I agree with Philippe about systems biology. They really need to develop this item. But I think we will face it, and we are trying to face it, and we, of course, should do it. And the loss of historicity, it's true. It's something which is, I think, very universal, unfortunately. And pressure to publications, etc. And then you forget the first basic classical experiment. And I think it's our role as teacher to put the things in perspective and not to try to make too complicated things, but put very simple things in perspective, and that they enjoy to learn this.
So I want just to add a few questions, and I hope that then there will be really a discussion, and this is just to add some more things that I have faced during my 30 years of teaching, and I believe that we face a problem regarding knowledge that we want to teach and disseminate, if it has to be more and more focused or eventually more and more broad. And this is quite important because, of course, to be very broad sometimes means also to be superficial. And we know that many things right now are becoming very superficial. And so to make decisions about to teach in a very focused way and or in a very broad way may have consequence. Then, of course, the other topic which I think it's key is should we stick to our discipline? And I remember that I tried very much to link up with microbiologists, virologists, and to have more people, around our own discipline, immunology. And it was always very difficult to really establish a real interdisciplinary course. And this is probably because of the languages. We don't speak very often the same language, and immunology has built a language on its own that very often prevent people from even reading our papers. Then what we have discussed in these 2 days, should we train people more to do mouse immunology as we have been doing for the last 40 years, or should we start really seriously to think about human immunology and the human immune system, which, as you know, is much more difficult to publish because you cannot do experiments in humans, and therefore you face the same question that have been raised regarding publication, etc. And then the other 2 points I think maybe we can discuss is about what kind of training we want to provide to these PhD students. Generation of data, so experimental work, or maybe in 10 years or 20 years, there will be no need to generate any data. It will be only about data mining and data management. Now, these topics, we have to, have a vision and decide quite soon if we want to teach this, because it's a whole discipline in itself. And then, of course, complex system. The immune system is a complex system, and clinicians, they know pathology are very complex, and they can certainly teach us a lot. Diseases are very complex, and they try to tackle this topic. But I believe that so far we have not tried to do much in this regard. And then finally, I think we have also some kind of social responsibilities. For instance, to really discuss if we want to do more basic research, even more, or maybe it's time to go to more applied research, meaning translational, because we have to deliver to the society. Maybe something, out of our knowledge. Do we need, as Philippe said, more PhD students or less? Maybe we are just training too many PhD students. This is an important issue because people have done calculation on the output from China and India and Southeast Asia, and it's quite a frightening number that maybe we will do something that at the end we'll have no possibility to ever to do research. And then, as you know, there is a lot of interest in social networks, and we normally don't really use this very much. I think that maybe we should think about the usage of social network for dissemination of knowledge and not only pictures. And then my final point that I can't see, but maybe I remember, is that maybe we also have a responsibility about the dissemination to the public, because again, in immunology, we don't really spend much time to transfer these, to transfer some of our discoveries, for instance, vaccine and benefits of vaccine to the public, and this results in a public perception which is that we are useless. And I believe that maybe now it's time, through the young generation, maybe to also ask the question if we do enough to reach the public and maybe also the politicians. Okay, that's what I wanted to say.
Okay, first I want to say that I didn't coordinate anything with Philippe, but what I'm going to say sometimes is quite reminiscent of what you introduced, so that's fantastic. So, today this is Philippe Kourilsky Jubilee, in fact, right? And we had a precedent a few years ago, not far away, just across the street, Pastors Jubilee, and it's basically the same setting, and the only difference is that Philippe is still walking by himself, and President Sarkozy is not here in the room today, while Sadi Carnot was in fact at that inauguration. But beware, Philippe. Philippe, in '92, Louis Pasteur Jubilee celebrated his most important contribution. That was the germ theory of disease. And at the very same time, a totally unknown guy was proving him wrong in Russia, okay, by discovering the first virus. So probably now somebody is proving you wrong, something that you did about 10 years ago, and it's probably totally disproved at the time. I like this story because this is such a simple experiment, you know, take the juice of an infected tobacco leaves, filter it onto the Chamberlain filter, take the juice back, put it on a healthy plant, get the disease again, okay? This is incredibly simple, okay? And why is that a fantastic experiment? This is a fantastic experiment because it is very simple, but it is important because it is a conceptual experiment, okay? Concepts are at stake here, okay? And so I think this is a very important idea that we should try to convey to the young generation, that all experiments are not the same, okay? Some experiments are much more important than others. However, to do that, you must know what a concept is, and actually the notion of a scientific concept is not that clear, and it took 10 or 11 or 15 different schools of thought in epistemology in the last century to reach a total disagreement about it. So in the next 1 or 2 hours, I will do a quick survey of modern epistemology, okay? I call that Epistemology 101, okay? Well, I was able to summarize the work in one sentence for each of the schools, basically, okay? So first you had the Vienna Circle, for which science is the sum of basic empirical facts, okay? Then you have Karl Popper, for which science is what can be proven wrong, at once. A definitive experiment, the falsification story. Thomas Kuhn, science doesn't move much until it jumped in your face. This is the revolutionary way of looking at paradigmic changes and shift. And Paul Feyerabend is the anarchist, okay? All the above is wrong, anything goes, okay? And now, if we can, some Frenchies, I like Bruno Latour. Bruno Latour, for which, you know, science is what scientists do in their laboratory with a goal in mind. Whatever, we don't know what they are looking for, but they have a goal. And I love, my favorite is Jacques Monod. Monod was the best, you know, advocate of Karl Popper. Science is what is recognized as a valid endeavor by most scientists at a given point in time. Okay? And the French translation is just below. I must say that during my whole career, I've been a fervent adept of all of those different point of views, and they all contradict each other. And I'm not talking about the worst of all, Ludwig Wittgenstein, the one of 2 books that nobody can read because this is so unreadable. Actually, the 2 books totally contradict each other, okay? But it's always quoted as the most influential philosopher of science of the last century, by the philosophers, of course. But I will retain one thing that I very much love from Ludwig. This is the duck rabbit, okay? And I will let you look at that picture a little bit, okay? And he used that picture to illustrate the difference that we have to do when we use the word seeing or when we use the word interpreting. There is no seeing without interpreting, okay? And I'm using this fantastic figure, which in fact is not from him, he took it from a psychologist of his time, that in fact science can also progress by the introduction of new concepts in the context of exactly the same amount, the same context of data. The same data, like here, can be interpreted into totally 2 different ways that are totally unreconcilable, actually, totally inconsistent. So this actually, this is very interesting. Well, and I will illustrate this concept to show that it has, like all the other school of thought, a very operational value at one point in time in your career. With the MSc concept, the restriction, the MSc restriction concept, okay? In the pre-96 era, okay, the phenomenon of MSc restriction was very well established. Actually, he gave the Nobel Prize to Doherty and Zekernagel, okay? The MSc antigen was the signature of the self, and that was the Nobel Prize for Snell and Dosey. And the MSc was a polymorphic ligand that was recognized by a receptor, okay? So now let's do a little bit of rabbit ducking on this slide. This is the type of figures you had in all the most advanced, I would say, publication of the time, okay? You can see the T-cell receptor, and you see the way the T-cell receptor is, in fact, interacting with both the MHC and the antigen in a restricted manner that nobody could understand. Well, let's duck rabbit the whole thing. With the peptidic self model that I had the honor, luck, and big pleasure and fun to propose with Philippe a long time ago, we totally reverted exactly in the duck rabbit way, the whole thing, by making the MHC not a receptor, not a ligand anymore, not something that was recognized, by making it into a receptor of peptide, okay? There were a lot of difference with the view of other people before. And also, something was strange because this was the first receptor that was, in a way, non-specific. It has to be selective, but non-specific. So it was a new kind of receptor because it was not a key lock model, okay? Because the receptor has to be absolutely specific, okay? And the restriction part was not now a phenomenon that was due to the T-cell receptor recognition, but was due to the selection of peptides, okay? And then the new figure was this, and this is taken from the original paper, from which there is no electronic form at all anymore, and I don't have any, and this is taken from the, and of course, thanks, Pamela Bjorkman proved us right 1 year later, okay, with that beautiful historical figure of hers, of the structure of the thing. So what do I want to say? What is the axe I want to grind now? I truly believe that long-term sustainable economic growth depends on paradigm changes, not on hectic innovation, the way we are doing research at the moment. Okay? Think about George Boole, an aristocratic reverend that was living in 1870, I think, okay, who had the silly idea to reprogram, to rethink all math and logic into 0 and 1. You say, well, we must be able to do that, okay? And he did this work on this. And then Turing, Alan Turing, finished to work in the 40s, 50s, okay, who had the fantastic idea that up to now, all machines are specific tools. They are specific, they are dedicated to a given task. Can we think about a machine that will be a generalist machine, that will do anything, and achieve that by separating the program from the execution, okay? And all the modern economy is based on the research, totally silly research of those two guys that were working 70 years ago, okay? And this is the very last leap forward that we do in economy, in the Occidental economy at least, okay? And since then, I don't think we've done anything that ambitious, okay? And I really think that this is the reason why our side of the world, that was our genius, I think our side of the world is dying because we haven't done, or we haven't this kind of research for a very long time now. And the next step is to try to reinvest this type of science and to try to promote around the young people this type of research. For this, they have to know what are the good experiments, they have to know what is a concept, and I think we have to tell them epistemology.